Okay, now we've had a chance to look at a gene page and look at a transcription unit. Let's take the time to look at the Genome Browser. I'm going to click through to the gene argc. Here we are in the argc gene page, same basic layout as the argb gene page we were on earlier. And I'm going to go here to this line, the Click for Genome Browser line, and click on it. And this takes me to the Genome Browser. There's a Genome Browser for every one of the organisms in the BioCyc collection. The Genome Browser shows you, to scale, a portion of that organism's genome. If an organism has multiple genetic units, multiple replicons, for example, a microbe that has 2 chromosomes or a chromosome and 2 plasmids, you'll actually just go to the single one that your gene of interest is on. And you can, if you remember from part 1, the BioCyc query page, choose and go directly to one of the Genome Browser views for that organism without having to start from a gene page at all. And as I noted, the Genome Browser is to scale. So this little red area up here, which I'll mouse over right there, shows the actual chunk of the chromosome that we're looking at right now. And it will move around and it will become larger and smaller to reflect how much of the chromosome we're looking at and which part we're looking at. In this Genome Browser view, all genes of the same color are in the same transcription unit. Genes in the same transcription unit also have this gray shadow box behind them. And you'll notice as I mouse over things that I'm getting the gene's name. When I just go over the box, I get the transcription unit. So this tells me I'm in the FSA-B GLDA PTSA transcription unit. When I go down here, it shows me that I'm over PTSA, and it tells me what its product is, part of the PEP phosphotransferase system, and it shows me the intergenic distances. That is, how far is it on the genome from both of its neighbors. You may also notice that genes have different shapes at the end. We have a little legend up here that explains what you're looking at. A little triangle like this is a protein coding gene. A sharp angle off like this is an RNA coding gene. We actually show terminators, transcriptional terminators when they're known. We have transcription start sites, which you can't see right now because of the level of detail we're showing, but we can actually zoom in and out using this handy control to the left. So for example, if I want to see a little bit more detail, I can zoom in once, and we actually have a little bit of a zoom in and a larger zoom in, and I'll use the larger zoom in. And I'll do that one more time. And now you can see transcription start sites, all the little arrows, and if I click on them it will take me to that operon. I'll also point out up and to the right that our red band has narrowed because we've zoomed in. We're now looking at a smaller portion of the genome. Now say we decide to zoom out again, once, one more time, and I'm going to keep zooming out another time. Now you can see everything's squished because we're looking at stuff in scale. It can actually make it a little hard to represent the genes at this level. And I can also navigate left and right. So we'll zoom back in so we can actually see some gene names. And mouse over, it's easier to see where things are. I'll zoom in one more time. And now we're back to seeing the gene names again. And now I can just mosey on over to the left. This is arbitrary left. I'm just going down in numbers on the genome. And now I'm looking at a different chunk of the genome. I can see different transcription units. And say I decide I want to click on one of these genes. I'll click on METF. That takes me to that transcription page. I can also go back and click on the name here, and that will take me to the gene page for METF. And then what we can see is if I go to the gene page here for METF and click on the Genome Browser again, it puts me back in the Genome Browser, but now METF is the gene I'm centered on. And you can always tell what gene you came to the browser from and what your sort of current gene focus is because you'll have these little hatch marks across the gene in addition to any coloration for operons, transcription units, etc. Now let's say we decide that we've strayed too far afield from our gene in biosynthesis and we want to look at another pathway gene. Say we want to look at ARGD. I can go to ARGD within the Genome Browser by going up to this gene name area, typing ARGD, and clicking Go. And now I'm centered on ArgD. We can see the cross-hatching down here again. I move over it. It tells me what pathways it's involved in. It tells me what its product is, intergenic distances, product name, all that stuff. And I can click on ArgD, and it takes me to the ArgD gene page. So now that we're on the ArgD gene page, I'm going to take a moment to explain the gene reaction schematic. So let's mouse so that it's right in our field of view. So here, get the name here, is the gene reaction schematic. Again, there's a white question mark and a green circle to explain this, so you don't have to remember my explanation. As I mentioned toward the beginning of this part of the introductory series, the gene reaction schematic shows the relationship between genes, proteins, and the reactions those proteins catalyze. It can also show the relationship between genes and RNAs in the case where a gene codes for what we'd call a terminal RNA, so rRNA, a tRNA, something where the RNA is the final product. You'll notice we don't explicitly represent mRNA at the moment. In the gene reaction schematic, the purple boxes are genes. In this case, the thing that you're currently on is highlighted, and since we're on the ArgD gene page, like this, that's highlighted. So purple boxes are genes. Then the orange circles, or perhaps the mustard circles, are protein products of those genes. So here we see that the product of ArgD is the ArgD monomer. Now we have a little 2 and another circle, and that shows us that the ArgD monomer becomes part of a protein complex, in this case a homodimer, and that homodimer is the bifunctional enzyme N-succinyl diaminopimylate aminotransferase / acetyl-ornithine transaminase, which is quite a mouthful. And because it's a bifunctional enzyme, this then hooks up to 2 different reactions, which is what these blue boxes are. When possible, reactions are labeled with the EC number for that reaction. We also use partial EC numbers, which would be in the form of 2.6.1. When we know in general what EC category a reaction should fall in, but no official EC number has been assigned yet. And you'll notice as I mouse over things, it tells me what I'm over. It tells me that I'm over the protein ArgD. It tells me I'm over the bifunctional enzyme here. If I go up, I learn that I'm over AST-C, or its own bifunctional enzyme. And as I go over the reactions, I get a concise description of the entire reaction so that I know, without clicking on it yet, that this is a conversion of N-succinyl LL2,6-diaminopimylate to N-succinyl 2-amino-6 ketopimylate with a conversion of alpha-ketoglutarate to L-glutamate in the process. Again, quite a mouthful. And we can actually use the gene reaction schematic to click through from item to item. So for example, I'm going to use this to lead into the next and last portion of this Introduction to BioCyc webinar series, where we're going to look at proteins and reactions. So we're on ArgD right now. I'm going to click through, and now we're on the enzyme page for N-succinyl diaminopimylate aminotransferase / acetyl-ornithine transaminase. And this is where we're going to pick up the 5th and final part of this Introduction to BioCyc series, explaining how proteins are represented, what kind of information we have for them, and how we show reactions. So I encourage you to come back, round out your Introduction to BioCyc, and get yourself ready to use the BioCyc collection in earnest. Thank you.